Next speaker is Dr. Wang from Ludong University in Shandong. His presentation is a research model and its evaluation of seahorse bacterial enteritis.
Thank you, Dr. Zhang. Good morning, everyone. It's my pleasure to give a speech here. My topic of speech is research model and its evaluation of seahorse bacterial enteritis. Here is the outline of my speech. In China, agriculture of seahorses have been very popular in the last decades. And now there are hundreds of seahorse farms along Chinese east coast. Well, seahorses are very strong to stresses, especially under farm conditions, which make them more vulnerable to disease. Here is a question: how many types of disease have been reported, and which disease has most impacts on seahorse agriculture? So we researched and analyzed all the scientific publications from 1951 to 2020, and we can see from here all these references can be grouped into four groups according to different habitats of the seahorse disease occurred: aquarium, farm, wild, and lab. And in entering this century, the number of these publications increases 3 times. And next, we look into the global distribution of seahorse diseases. For now, there are 23 countries at least contribute to 1 publications to the world. China contributes most and accounts for 34% of all references, especially in farm conditions. And then we look into the seahorse species affected by different types of diseases. And we can see from the left panel, Hippocampus erectus and Hippocampus cauda are the most affected seahorse species and count over 15% of all publications. This may be due to the agriculture in China. And in the left panel, there are 11 types of diseases has been reported, and we can see in the and they are generally divided into 2 groups, and we can see in the purple circle. The first group is infectious disease, and they count about 80% of all kinds of diseases, and the rest of this and other diseases belongs to non-infectious diseases. And in the infectious diseases, bacterial disease accounts for 34%, and it's the most. And for now, about 30 bacterial pathogens has been identified, and most of these pathogens can induce enteritis and cause heavy losses in seahorse agriculture, as said before of Dr. Zhang. So we began to look into the bacterial-induced enteritis in line seahorses. And first, we isolate and identified a new bacterial pathogen by gram staining, and SEM sequencing, and biochemical analysis, and confirm its pathogenicity. And here comes another question. If we want to review the pathogenesis of the seahorse bacterial enteritis pathogen, what is needed most? There's no doubt a reproducible experimental model is needed. So we are going to build a research model of bacterial-induced enteritis in seahorses. We use the different dose of challenge of Rosella tartar to challenge seahorses, and we can see the representative annual inflammation of enteritis showed up in D3, and it become more severe in D14, and then it restored in D21. After dissection, we can see in the intestine, the content became transparent, and there are a lot of fluid rotation in the intestine. After we build a research model, we want to evaluate. So we began to construct a scoring system for evaluating the model. We firstly select 44 external histological and molecular parameters, and finally find 19 of them useful. And it should be noted the score of each parameters cannot exceed 4 to avoid overweighting of a single parameter. And next, we look into the variation in the detailed parameters. The first is body length and weight. By Rosella tartar infection, the body length increase was suspended, and the weight was decreased in both challenged group. Next, we look into the variations in intestinal tissue. There are 3 variations. The first one is number of goblet cells decreased in D1, and gradually increased from D3 to D14, and decreased again in D21. The second is the infiltration of inflammatory cells in the mucosa was observed from D1. And the last is the thickness of both laminar propria and muscularis mucosa of the intestine gradually increased from D3 to D21. And then we determined the gene expression of 6 intestinal immunocytokines, including both pro-inflammatory factors, interferon-1, interleukin-1 beta, interleukin-1 beta receptor, and TNF-alpha, and as well as anti-inflammatory factors, interleukin-2 and interleukin-10. We can see the gene expression of the 6 intestinal immune cytokines share a similar variation trait as the histopathological result. And the gene expression for intestinal antimicrobial peptides are the same as that of the immunocytokines. They all share a variation pattern here to have a significant increase in D14 and then decreased. Interestingly, we found that TOR5 gene expression is very different from other genes. It significantly raised in D1 and then decreased, and then raised up even higher than D1 in D14, and then restored. So we speculate a key role of TOR5 in mediating the immune response of the host. In detail, Rosella tartar infection can trigger TOR5 signaling to stimulate a wave of mucus secretion and induce the suicidal apoptosis of goblet cells to expel pathogenic bacteria at D1, and then active pro-inflammatory genes expression to mediate intestinal inflammation pathway, to mediate intestinal inflammation, the differentiation of goblet cells and the proliferation of inflammatory cells, active pro-inflammatory pathways, and in turn upregulate both anti-inflammatory pathways and intestinal gene expression of antimicrobial peptides in D14. And thereafter, the intestinal inflammation generally relieved until D21. So based on the evaluation of all 19 parameters, we finally established a research model and evaluating system which can factually reflect both of the pathogenic mode and the intensity of bacterial enteritis. Here's the highlight of this research. Edward zeta tartaralis novel chronic enteritis pathogen in farm cultured land seahorses. An experimental model and its evaluation system of bacterial enteritis in seahorses was successfully established. Top 5 may have an essential role in triggering host immune responses. And this model can be used for revealing the pathogenesis of bacterial enteritis, identifying potential therapeutic drugs, and searching suitable genetic markers for seahorse molecular breeding. Here, we want to thank to our cooperative partners and our research team. Thank you for your audience. Thank you very much.
All right, thank you, Dr. Wang. Any questions?其实这个海马养殖最大的问题就是病态，就是病态。这个病当然跟我刚才讲的，跟那个青海马的质量有非常大的关系。因为我们国家现在养这个海马，现在最大的 challenge 就是这个种源，是不是？Okay, any question, online friends?